Good morning, good afternoon, good evening. I hope this presentation is finding you well, and I hope that you are striving during these hard times in quarantine. My name is William Harrison, and today I will be discussing more about neurofibromatosis 2. Hopefully you guys will remember some aspects of NF2 in my previous presentation, but I would like to just reintroduce some of the main topics so you guys can become familiar with it again before I discuss some of the biochemical aspects of the disease, current medications, novel targets, as well as areas of future research. So, as previously stated, neurofibromatosis 2, otherwise known as NF2, is a tumor suppressor gene on chromosome 22q12, and this gene encodes a protein named myelin. Now, myelin is known to affect multiple cell signaling pathways. One of the leading causes of NF2 is a constitutional mutation in the NF2 gene. This is known to cause an autosomal dominant disorder affecting about 1 in 33,000 people, and this is characterized by the development of tumors primarily affecting the nervous system and a lot of different areas. Now, approximately half of the cases known are due to de novo mutations not inherited from a family member. NF2 mutations in neurofibromatosis 2 can either be due to germline or somatic mosaic, with the latter referring to mutations present in only a subset of cells. Now, something that I failed to discuss previously that I think is very important for you all to understand is that NF2 follows what is called the Knudsen two-hit hypothesis. And for those who are unfamiliar with this hypothesis, otherwise known as just the two-hit hypothesis, is that most tumor suppressor genes require both alleles to be inactivated, either through mutations or through epigenetic silencing to cause the phenotypic change. Specifically with myelin, typically what is seen, as previously stated, is a constitutional or germline mutation occurs, initiating the first hit. And an additional somatic mutation, otherwise known as the second hit, being required for the loss of heterozygosity and tumor suppressor inactivation, followed by tumor formation. Now, the majority of tumors associated with NF2 are otherwise known as schwannomas, meningiomas, or epineumomas. For this presentation, I will be just focusing on a specific type of schwannomas, otherwise known as vestibular schwannomas. I have a couple reasons for this. One is primary literature is actually focused on treatments that are specifically removing vestibular schwannomas, as well as a lot of current treatments are actually only doing some of their cell lines based on cell schwannomas. So I feel like it encompasses a majority of what NF2 is, and so there's no need to vary off into other realms. Now, moving forward, NF2 somatic mutations have also been found in a multitude of other cancer types, which is very nerve-wracking, if I must be completely honest. And this is not just limited to the ones I'm stating here, such as mesothelioma, anaplastic thyroid cancer, breast cancer, or liver cancers, but it can be found in others as well. Now, missense mutations are more frequent in cancer than in NF2, and they occur in only a small subset of patients with NF2. This is partly due to the lack of predisposition in developing cancer in patients with NF2. Though the explanation is not complete, since there remains considerable overlap between NF2 and cancer-related NF2 aberrations. Now I want to move on into the molecular, biochemical, and physiological aspects of NF2. To begin with, I want to describe the NF2 gene as well as some of the symptoms that are caused by the schwannoma that is developed by the lack of the tumor suppressor gene. So moving forward, the most common tumor, as I previously noted, with NF2 is called the bilateral vestibular schwannoma. Now, initial symptoms of NF2 in regards to this schwannoma are hearing loss, facial nerve impairment, visual disruptions, and skin tumors, with the majority of patients presenting one or a multitude of these symptoms before the age of 20. Now, the NF2 gene is known to contain 17 exons, and its product, Merlin, is a tumor suppressor, which controls cell signaling pathways implicated in cell growth, proliferation, as well as morphology. Now, additionally, Merlin acts as a membrane skeleton scaffolding protein that localizes underneath the plasma membrane at cell-to-cell junctions. It is known to link the cytoskeleton to the cell membrane. There are actually two isoforms of Merlin. One is type 2 and the other is type 1. Type 1 includes exons 1 through 15 and 17, and typically with type 1, the exon 16 sequences are actually inserted into the extreme carboxyl terminus of the protein, while the type 2 Merlin does not have any existential features that differ from the Merlin itself. Now, what is really important to understand, and the reason why I have the visual right next to the text, is showing how Merlin is activated. Now, Merlin is continuously phosphorylated, which means that this phosphorylation is inactivating the protein. Thus, it requires what is called a phosphatase, which is an enzyme which is able to directly activate Merlin by removing or dephosphorylating it. It is known that the inactivation, otherwise the phosphorylation of Merlin, contributes to the malignant conversion in multiple cell types, as does the loss of Merlin expression because of a mutation. Importantly, phosphorylation not only causes the conformation of Merlin to change to open and active state, but it also targets Merlin for polyubiquitination and degradation by the proteasome. The decreased ability of mutant Merlin to effectively act as a tumor suppressor, at least in part a direct result of the decreased half-life of mutant compared to wild-type Merlin. Now, there are several binding sites in the Merlin protein that are crucial for Merlin's function, being critical to interactions with other proteins or for correct Merlin folding and activation. Now, Merlin must associate itself in order to change conformation and to, become active. This is achieved first by the folding of the N-terminal domain, which involves the interactions of amino acids 8 through 121 and amino acids 200 through 320. After this conformational change occurs at the N-terminus, Merlin can then transition to what is known as the active or closed conformation by the association of the N-terminal amino acids with the C-terminal amino acids. Now, when the phosphatase, otherwise known as myosin phosphatase, is in somewhat of a similar location as Merlin, it is responsible for the dephosphorylation at residue serine 518, and it interacts with the Merlin via its subunit at residues 312 and 341, specifically at Merlin's exons 10 and 11. Now, this region has been shown to have multiple missense mutations in the NF2 patients. And this is going to be further researched as to why at these residues are we having missense mutations. Is it just due to randomness, or is it due to something else? But this gives you an idea of how the interaction occurs between the phosphatase and Merlin. Now, P21 activating kinases are regulated by Merlin, as seen in the photo to the right. And they also regulate the activity of myelin by phosphorylating at that serine 518 residue. And this causes myelin to change to the open, or otherwise known as inactive state. Now, PAC, or P21 activating kinase, will interact with myelin at the N-terminus between amino acids 1 through 313, and there are many known missense mutations in NF2 found in this region. Additionally to all this information, unfortunately I wasn't able to find a graph that showed all of these, but a protein, otherwise known as PIKE-L, will bind to myelin in the region containing amino acids 1 through 332. Whereas mutant myelin harboring the L64P missense mutation will not bind to PIKE. Importantly for this, myelin cannot suppress tumor genesis, including cellular proliferation via the inhibition of the P13K pathway without PIKE-L. Myelin's interaction with PIKE-L could also regulate downstream effectors in other signaling pathways such as the AKT and mTOR. Thus, this missense mutation is very important at the L64P. And something really interesting is this mutation has not been found in any other cancers except NF2. Just kind of a fun fact. Moving forward in the presentation, I would like to discuss a lot of different signaling pathways that are crucial to cell proliferation that are actually inhibited by myelin. Some of them I've previously discussed, but I'd like to go into a little bit more detail with some as well as introduce others. Now, the main few that I will be discussing in this are the PIKE-L, the mTOR, the SARC-FARC, otherwise SARC-FAC, sorry, the ERK1 and 2, as well as the PDGFR and the RAC-PAC. Now, beginning with the P13K PIKE-L, the P13K pathway is activated in a multitude of human cancers, and it contributes to increased cell proliferation and metabolism and decreased apoptosis when activated. And myelin is known to be able to suppress this pathway by binding with the P13K enhancer isoform, hence preventing this pathway from being activated. So in individuals that have this mutated myelin that is not functioning correctly, this pathway can remain active, and one of the causes of the continuation of cell proliferation as well as the development of tumors. Moving forward, we have the mTOR1 pathway. Now, inhibition of the mammalian target of rapamycin complex 1, otherwise known as mTORC1, due to myelin activation leads to the inhibition of mRNA translation, which causes an increase in apoptosis, limiting cell survival and blocking tumor initiation. Now, mTOR inhibition is accomplished through multiple pathways, including the AKT, ERK inhibition, and integrin-specific adhesion via the PAC. But mTOR has also been identified as an important pathway in meningioma and schwannoma cell growth. So again, just showing another aspect of how important myelin is in inhibiting all of these specific pathways. Moving forward into the SARC-FAK Paxilian pathway, myelin is known to be able to bind to receptor ERBB2 as well as SARC, inhibiting SARC from binding to and being phosphorylated by the ERBB2 in a competitive manner. As a result, the SARC cannot activate focal adhesion and Paxilin, which inhibits cellular proliferation and growth. Interestingly, binding of myelin to the ERBB2 is initiated by Paxilian binding to myelin on exon 2, causing myelin to move to the plasma membrane where it can then bind to ERBB2. Myelin is also known to inhibit FAC from binding to SARC and P13K. Now I will continue this presentation in a second video. Unfortunately, the software that I use won't only allow me to video for about 15 minutes.